Graduate Diploma in Bioinformatics, Madurai Kamaraj University from 2006 to 2007. PhD Biotechnology, Department of Biotechnology, Allahabad University, Karaikudi from 2009 to 13. UGC Dr. D.S. Kotari Post Doctoral Fellow, Department of Microbiology, Bharatidasan University from 2014 to 15. RUSA PDF Postdoctoral Fellowship from RUSA Phase 2 from August 2019 to March 2021. Ma'am has totally 14 years of working experience, worked as assistant professor at Department of Biotechnology at Dr. Umayal Ramanathan College for Women from 2021 to till date, worked at as RUSA PDF at Department of Biotechnology, Alagappa University from 2019 to 2021. Worked as a scientist at Satyabama Institute of Science and Technology, Chennai from February 16, 2015 to March 31, 2019. Completed one PhD student. Worked as a postdoctoral fellow at Department of Microbiology, Bharatidasan University, Tiruchi from 2014 to 2015. Worked as CSIF SRF at Department of Biotechnology, Alagappa University, Karaikudi from 2009 to 2014. Ma'am has research experience, worked as scientist, C Center for Nanoscience and Nanotechnology, Satyabama Institute of Science and Technology, Chennai from 2015 to January 2018. Worked as UGC Dr. D.S. Kothari Postdoctoral Fellow at Department of Microbiology, Bharatidasan University, Tiruchirappalli, Tamil Nadu. Worked as Senior Research Fellow at Department of Microbiotechnology, Allahabad University, Karaikudi. Worked as Research Associate at Department of Biotechnology, Allahabad University. Worked as Research Scientist at Institute of Informatics International Tech Park, Bangalore, India. Ma'am has awarded Excelsior Award, RUSA PDF Postdoctoral Fellowship from RUSA Phase 2, Certificate of Appreciation in Recognition of Research Publication in High Impact Journals from Satyabama University for outstanding contribution in the area of functional genomics and bioinformatics from Young Scientist Startup Grant Award, Dr. D.S. Kothari Postdoctoral Fellowship awarded by UGC India, Overseas Travel Grant awarded by Department of Biotechnology, Government of India, Senior Research Fellowship awarded by Council of Scientific and Industrial Research, New Delhi, State Eligibility Test for lecturership in Tamil Nadu conducted by Bharatiyar University, Government of Tamil Nadu, Scholarship for Advanced Diploma in Bioinformatics, Certificate of Merit for securing third rank in BSc Zoology, examination of Madurai Kamaraj University, and Dr. M. Varadarajan Memorial Endowment Prize, Academic Proficiency Award, First Prize College Topper, Alagappa Government Arts College, Karaikudi. With this short note, I welcome ma'am to present his session. Welcome, ma'am. Now the time is yours.
Thank you, Nandini, for your brief introduction about me. Thank you very much. And first and foremost, good evening to all. Hope this session finds you beneficial for you all in doing your research. So let's start up with the session now. So you all can see my slide, right?
Yes,
If not,
ma'am.
you can let me know.
Yes, ma'am. Yes, ma'am. It's visible, ma'am.
Thank you. Thank you, Nandini. So we are into the 2 days training workshop series. So I'm going to handle molecular docking procedures. So I have separated the session into two. One will be the theory part. Today we will be having the theory part, and tomorrow we'll be having the demonstration using a tool. So in the theory part, we will cover about the basics about what is molecular docking. So general introduction about molecular docking. Then we'll see the terminologies which is being included in molecular docking procedures. Because when you read a research article, you can find so many terminologies, right? So we will be confused what types of terminologies, because I was confused when I was a researcher. I hope I can clarify with you if you have the doubts. So we'll see about the terminologies which are being involved in molecular docking. Then we will see about what are all the applications where you are going to apply molecular docking. Because molecular docking is the heart for the computational approach. So wherever you are going to apply it, molecular docking plays a major role. So, if you're being a researcher or a student, you're going for an internship or you're going to design a project work, you're supposed to know about this molecular docking. It will help you to validate your results. If you have your experimental data, you can validate your results with the molecular docking procedure. Or in a vice versa situation where you have your computational data from the molecular docking approach, you can take it for the experimental approach and you can validate it. So vice versa, both the ways molecular docking procedure is going to be beneficial for you as a researcher. Then after seeing the applications, we will see what are all the steps involved in molecular docking and what are all the tools which are being currently using AI tools, web servers, standalone tools. We can see all those one by one in this session. Okay. So the first introduction for molecular docking is, so molecular docking is a computational technique. It is used in structure biology widely and drug discovery process. So in structure biology, say for example, you have a protein structure, you can see that this protein is involved in a particular type of disease. On interaction of a protein with a ligand, you can understand the structural basis or the biological process of this particular protein. So they can either inhibit the process, biological process, or enhance the biological process. So from this figure, you can see that a ligand is interacting with a particular site, that is the binding site, and there are so many interactions. These interactions reveal that they have an atomic level interactions. So atomic level interactions is when you have a ligand. Here in this picture, it is clear that you have a ligand. It is having an oxygen atom interacting with the cysteine residue. It means that the oxygen showing an interaction with cysteine amino acids will reveal you the atomic level interaction. So we can understand the atomic level interactions of your study. So the general process involved in molecular docking is it can predict how a ligand can interact with a macromolecule. So ligand here, it is a small molecule and macromolecule is a protein here. So they, on interaction of a protein with a ligand, they form a stable complex. This stable complex will either inhibit the biological process or enhance the biological process. The same theory is being studied in biochemistry in enzymes chapter, where you have a lock and key hypothesis. So you have an enzyme where it interacts with a substrate, it forms an enzyme substrate complex and ends up with an enzyme product. So this product will either enhance the biological process or it will inhibit the biological process. So this product is going to be the content where the same theory is being applied for molecular docking procedures. And also another terminology is all enzymes are protein, but all proteins are not enzymes. So you have a terminology here as receptor and as a ligand. So what is a receptor and what is a ligand? So receptors are nothing but proteins. Where the ligand can directly bind to one target molecule. So the target molecule here, they bind to a specific site. This specific site of the target molecule, this target molecule you can name as receptor, enzyme, or metal ions, where ligands are molecules that are binding to the target molecule, right? So they are classified into further as small molecules, ions, proteins, and peptides. Small molecules are often they are called as organic compounds. They include drugs, hormones, and signaling molecules. So ions are here classified as metal ions, where they bind or they have an centralized position where they involved in various coordination complexes. So similarly, protein itself can act as a ligand. So we call it as protein-protein interaction. Or with peptides, peptides are short stretch of sequences from 20 amino acids to 25 amino acids. There are also longer peptides than that, but shorter peptides or small peptides can have interactions with the proteins and they have certain biological functions. So these are the different classifications of the ligand molecules. And further terminology here, it is the binding affinity. So when a ligand binds to the target molecule is nothing but the receptor, enzyme, or the metal ions. They have a terminology called as binding affinity, which you are going to see in your molecular docking procedure. So binding affinity is nothing but a measure of strength of the interaction between the ligand and the receptor. So this, when you say it is a high binding affinity, it means that the ligand is showing a stable complex. The stability of the complex is going to be high. So we have seen that if the stability is high, then the molecular functions are going to be in a regularized way. So the next terminology is going to be the specificity. A particular ligand should bind to a particular type of binding site. It should have a specificity to that particular binding site. Only then it is like a lock with a key. Then when the key is fitting correctly to the lock, it will directly open the door. So similarly, when a lock when the binding site it is going to directly bind with the ligand with a high specificity, then the biological process is going to happen in a suitable way. So what are all the biological functions is it includes the signal transduction, enzyme activity regulation, and cell-to-cell communication. There are also so many other biological functions, but these are the basic functions which are being addressed. So other terminologies to know about the compounds is compounds means when you are reading a research article, you can see as compound. Somewhere you can see as small molecules. So all these, how the terminologies are being defined. So lead compounds. So lead compounds you have, if you are coming across a research article with the terminology as lead compound, it means that it is showing a promising binding affinity to the target protein. So if a particular binding affinity is observed, then it means that this particular entity is can be used for further preclinical studies or clinical trials. So such molecules we call it as lead compounds. So mostly these lead compounds are natural compounds, is existing drugs or novel chemical affinities which are being coming out from the lab. So they use this compounds and they screen against a particular target molecule and they find whether they show any promising binding affinity to the target protein. So such molecules we called as lead compounds. Next one is the optimized ligands. So in optimized ligands, you can see that they are generally being modified and or they are being improved for their binding affinity as well as the pharmacokinetic properties for where they use medicinal chemistry approaches in order to optimize the ligands. So this type of ligands we call it as optimized ligands. Then you have drug candidates. What are all drug candidates? So drug candidates means already the compound is being tested in undergoing extensive testing and optimization, where they are showing very good efficacy, safety, and they have suitable pharmacokinetic properties. So thereby they are into the preclinical or clinical trial processes. Next is FDA approved drugs for repurposing. So FDA approved drugs, it means the drugs are being approved for a therapeutic indications, but it is going to be repurposing or repositioning. It means that the already approved drugs which is used for the some therapeutic means therapeutic diseases will be screened out using molecular docking procedures and it can be used for other types of diseases. So this type of ligands or the molecules, you can use it for molecular docking. Next one is natural compounds. So we know that in basic research, we isolate many compounds from plants. We isolate it from animals. We use it. We use marine environment. We use soil environment. We isolate so many compounds from natural sources such as plants, microorganisms, and their potential therapeutics are being investigated through molecular docking procedures where do an extraction, you get the compound through GCMS and you identify the compound from the library and the list of library compounds can be screened for their binding affinity through molecular docking procedures. So such type of compounds we call it as natural compounds derived from the natural environment. Next one is protein-protein interaction inhibitors. It means that protein 1 is interacting with protein 2. But interaction of this protein and protein can lead to one specific type of disease. So this disease can be controlled by designing a ligand and binding to this protein 1. And thereby the protein and protein interaction cannot happen. Such type of inhibitor, you call it as protein-protein interaction inhibitor. So the inhibitors or moieties which is blocking a particular mechanism. So such type of compounds are also being used for molecular docking procedure. So all these terminologies will be identified in the research article. So when you use a set of you are going for doing some molecular docking procedures, you can find so many types of terminologies present in the research articles. So examples for the ligands is drug ligand. So in drug ligand, you have aspirin. So aspirin, you already know that aspirin is having an binding affinity for, it is used for preventing inflammation and also for fever. But nowadays it is not being used, right? So this aspirin is a drug ligand. It binds to the enzyme that is cyclooxygenase 2. This cyclooxygenase 2, which is converted to the substrate arachidonic acid. Any problem? Okay, fine. So this arachidonic acid is going to be the substrate which is being converted by cyclooxygenase 2 into prostaglandins. So prostaglandins are lipid moieties which are being essential for the physiological process such as inflammation, pain signaling, and the regulation of blood flow in different tissues and fever. So when aspirin is consumed, it is a drug ligand. It binds or how the process is initiated is it acetylates the serine 530 residue, and thereby it prevents the conversion of arachidonic acid into their prostaglandin, and thereby it can inhibit the inflammatory mediators and it can reduce the pain, or it can reduce the inflammation as well as the fever. So this is the one very good example for drug ligand. So next example for the ligand is the metal complex ligand. So metal complex ligand, we have two ligands. One is EDTA and other one is heme. So EDTA, you know that it is also called as hexadentate ligand. It has 6 sites where it can donate lone pairs of electrons. So this EDTA is used in chelation therapy where a human is consumed with heavy metal poison. In such cases, EDTA will help in chelating the metal ions by donating the lone pair of electrons, and thereby the poison from the body can be removed. So this type of ligands, you call it as metal ligands, where they have the metal center to interact with the lone pair of electrons. Similarly, in heme is also a porphyrin ring moiety where it has an iron group at the center which coordinates with the binding of oxygen. So oxygen binding to the iron molecule, it assists in the movement of oxygen from one place to another place in the body, and also in myoglobin it helps in the storage of oxygen in the muscle cells. So these two are the metal complex ligands, EDT and heme. Next one is the protein ligand. So protein ligand, here it is going to be the protein is here the hemoglobin, and ligand is going to be the oxygen, where it binds the oxygen, and this oxygen can be transported throughout the red blood cells throughout the body, and it is being delivered, and it acts as a carrier protein. So such type of ligand moieties, you call it as protein ligands. So understanding what type of ligands you have to choose and what type of receptor you have to choose is very crucial, so that you can design your project work. So next you're going to see about the receptor ligands. So what are all the receptor ligands? These are the vastly used receptors which are being studied for varying diseases. So you know G protein coupled receptors, GPCR receptor. The example is beta adrenergic receptor and dopamine receptor. This dopamine receptor is being highly focused for the diseases such as Parkinson's and schizophrenia, where multiples of compounds are screened against this dopamine receptor, and where they are acting as a neurotransmitter or cellular processes in Parkinson's disease patients. So such receptors can be used as a drug target for studying in various disease conditions. Similarly, in protein kinase, the classification here, it is the example is the epidermal growth factor receptor, and which is involved in various cancers such as lung cancer, where it acts as a signaling molecule, signaling system, and as well as for regulation. It is also being widely studied as an drug discovery role receptor for screening of large number of compounds. Next is the HIV protease. It comes under the classification of protease. This enzyme is essential in cleaving the proteins. This HIV protease is being used for blocking protease activity. So screening compounds against this receptor is also beneficial. The tyrosine kinase receptor, the examples are insulin receptor and epidermal growth factor receptor, EGFR, where it is involved in cell growth, differentiation, and survival. So in cancer-based study, it is being studied as an attractive target. So similarly, this nuclear receptor, estrogen receptor, it is present in mainly focused in breast cancer, and androgen receptors. So these receptors are called as nuclear receptors, which is being studied in cancer therapy, where estrogen receptors are being targeted with a number of molecules, which is involved in regulation of hormones, and it has a major role in gene regulation. So these receptors are being targeted, and they are they are the major classes of drug targets. So cyclooxygenase enzyme, as we have seen, aspirin is being screened against this particular enzyme, and it is also involved in the process such as inflammation, pain signaling, and certain types of cancer. So targeting this is also one of a drug target. Ion channels. In ion channels, the example is voltage-gated sodium ion channel, where it is involved in epilepsy and certain neuropathic pain conditions. So this targeting this voltage-gated channel will help in major roles such as nerve signal transmission, as well as modulating the activity, and can act as a therapeutic effect. So this voltage-gated ion channels are being used for various conditions, and it is also a suitable drug target. The integrins, which are being involved in cancer and autoimmune diseases, are also an attractive target because they are playing a major role in cancer as well as inflammatory diseases. So targeting this integrins will be a suitable target. So these are the major targets which are being widely studied so far. And there are several other biological targets, receptor targets being studied, but these are the major targets which is being studied. So when you are going to design a particular project, you can understand what is a ligand, what is a receptor molecule, and how you are going to design a project work. So next we are moving on to the types of molecular docking. So types of molecular docking, there are two types of molecular docking. One is lock and key model. It is also called as rigid docking. So rigid means you have a protein as well as a ligand. Both are going to stay rigid. It means that when a lock is having an exact rigid position, the key will come and fit and it will open the lock. But whereas the specificity is going to be very crucial here. Only if it is specific, the lock will be opened. So, and also the complementarity of the ligand with the protein is also very, very important. So here it is going to be a rigid docking. You have the lock and the key, which does not depend on the movement between, or they don't have rotatable bonds or rotatable complex being applied here, but it will directly have the specificity and complementarity. Only then the process of binding will happen. So next one is your induced fit model. This is the second type of molecular docking procedure. In induced fit model, it is an example of a hand in your blouse. So you have a blouse, it is smaller in size, but when you leave your hand inside the gloves, it expands and it adjusts and fits to your hand size, right? So same flexible hand shape position you can understand. So this is your protein and this is your ligand, where the ligand as well as the protein both can be flexible, or either a ligand or the protein alone can be flexible, and somehow they undergo some conformational changes, and they are guide in the stability, and they use some energy in order to fit the ligand into the binding site. So in a biological system, generally if you see the proteins are having some flexibility in nature. So this flexibility is applied for this particular type of induced fit model, and this is also having some dynamic interactions, showing some dynamic interactions between the ligand as well as the protein moiety. So this is another type of molecular docking. Two types of docking we have seen. One is rigid as well as induced fit model. So we are moving on to the sources which are available with docking. So standalone is nothing but the tools which can be downloaded from the internet. You can install it in your computer and you can use it. So this is called a standalone tools. So you need not you need not require any internet connection for this particular process. So standalones, when even if you don't have any internet connection, you can just keep on doing your docking studies. So one such example is your Autodoc. Using Autodoc, this is a widely used software where you can study the prediction of small molecules with their proteins. And from UCF, you have DOC, and it is also from University of California, San Francisco. The program has been developed, and it is used for virtual screening procedures. That is, binding interactions can be studied, as well as the virtual screening can also be performed. Next is the UCF Chimera. It is also used for molecular docking preparation. Before going for a molecular docking, we will prepare the ligands and the proteins. So for that, you can use UCF Chimera, where dock prepare tools are available, and you have docking procedure also included in that particular Chimera tool. Next one is Autodoc Vienna. This is the upgraded version of Autodoc, where it is efficiency is higher and accuracy is higher. So next standalone tool is Surflex Doc. You can also download it, but here you can see that it has patented search engines for ligand docking. So this is one of the other type of completely different from other. So each one has uniqueness in their program model. So the next one is the Python prescription virtual screening tool. It is shortly called as Pyrex. It is a user-friendly. It is having a user-friendly environment where it integrates with Autodoc as well as Beema and other tools, and where you have a graphical user interface in order to use your docking procedures and experiments to carry out. Next one is Plexix. This is also available. It is designed by Biosol IT. It is also designed for the flexibility of both ligand and the proteins, as we have seen that flexible docking, that is both the ligands and the proteins will be flexible, and the docking procedures can be carried out using FlexX. That's why it is called as FlexX. So next one is web servers. Though if you are not able to download and install in your system and you are finding it difficult, you can use docking web servers. So these are the docking web servers you can use. That is SysDoc as well as ZDoc, ClusterFlow, and HatDoc. So you can all these have a common place where you have a web interface where you can submit your job, and you can monitor your job status. Then you can, if you provide your email ID, all the results will be coming to your email. So similarly, if you see in SwissDoc, you can submit your protein as well as your ligand, and then your results will be updated in your email. In ZDoc, you can do protein docking. Here, protein small molecules as well as protein docking can be performed. When you're going for such type of docking, when you want larger space for doing such type of protein docking, so ZDoc will be a very good place, as well as HatDoc. It is also essential for doing protein docking, protein peptide docking, as well as protein nucleotide docking. Cluster Pro, here also you can do online protein docking, protein peptide docking, and protein DNA and RNA docking. So for all these things, you can submit your job and you can receive the output result in your email. So the other docking web servers or docking server, it is also doing ligand protein interaction, protein docking, and you can it also provides a visualization tool for analyzing your docking results. Next one is CAPS dock. This CAPS dock is also doing a flexible protein peptide docking and protein docking. Some web servers they specify what type of docking you want, what type of options you can select. All those things if you give, they automatically do those types of docking. Next one is benchmark for docking server. This is also a web server where you can do the performance and evaluate the performance, where docking algorithms are being given for each and every steps and protocols. You can select what type of docking algorithms you want, and then you can do for a do your docking experiment. So AI based docking resources. So you can do now AI has entered into each and every place, right? So we also have to get advanced with that. One type of AI based docking is deep dock, where you use deep learning. They are using deep learning based method for molecular docking, and it is also using convolutional neuronal network process to study and predict the binding affinity of the ligand and the protein complex. So BindSurf, it is also using deep learning methods for designing and prediction of binding ligand sites with the protein sites. And then you have neuronal networks which are being trained for studying the protein structure data and to identify and characterize the ligand binding regions on the protein surfaces. You have DeepChem. DeepChem is also a protein, so open source library where you will be specifying not specify the molecular docking procedures, whereas it builds trains the deep learning models and it gives you the chem informatics stocks, and then finally you will be doing the molecular, you will analyze the molecular properties and their activities. So this is the virtual deep views and DSS and virtual screening procedure where you can do molecular dynamic simulations here, and it also helps to predict the ligand binding affinities. So these two are going to be a different thing. Before going to the molecular docking procedure, you have to identify the binding site. So binding site is the site where your ligand is going to fit exactly into the particular protein. So this binding site needs to be identified. Deep site is a one particular tool used using AI where it provides us the local geometry as well as the evolutionary conservation about the protein, and it determines the identical potential binding sites for the particular protein. So this deep site is going to be an important tool for identifying the binding sites of the particular protein where the ligand needs to be bound. Next one is the alpha fold. If you have a protein structure, then it is well and good. If you don't have the protein structure, then you have to go for an homology model, or you are supposed to design a homology model. So in that case, AlphaFold is one particular AI based model where you can do your molecular modeling, and then you can, before going for molecular docking, you can refine your structure using this. So AlphaFold deep site or can be used before pre-docking steps before you are going for the docking procedures. So next we are entering into the applications of molecular docking. So molecular docking, it is mostly used in structural biology as well as drug discovery, right? So in drug discovery as well as as well as in molecular docking, you can see a concept called as virtual screening. Both these two concepts use molecular docking. So in through basic research, you have millions of compounds. These millions of compounds can be cut down, that is 5,000 to 20,000. They are being taken for drug discovery process. So for drug discovery process, from there you have to enter into the preclinical research where you use molecular docking procedure, where virtual screening procedure is being applied, where you find the best ligand bind. Now you know what is the binding affinity, right? So the high binding affinity or the stable complex of the moieties will be identified. Such ligands will be taken for the next preclinical research. So you have totally 250 compounds being screened out or cut down from this large databases, and then this 250 compounds will be taken for the preclinical research, and then from that the past or the highlighted ones will be taken for further clinical research. That is totally five compounds or the lead compounds will be taken as the drug compounds. Next one is from the file which are passing out this clinical research will be taken for the approval of the drug by the FDA. So this is how a list of compounds from millions of compounds it is been shortened down using this applications. So drug discovery and virtual screening use molecular docking for cutting down the number of molecules. At these two stages, one will be the drug. This whole process you have drug discovery where you use virtual screening, and one of the part is your molecular docking to find the highest binding ligand to the protein moieties. So drug discovery and the developing timeline is being discussed here. You have millions of compounds. The stage 1 you have drug discovery, you have 10,000 compounds. Next, the stage 2 is the preclinical development. You have 250 compounds. For all this stage 1 and stage 2, it takes around 3 to 5 years. So vastly, if you see that it is a timeline for a PhD candidate. It takes you take a particular sample or a natural compound. You identify millions of compounds from there. Then you go for a drug discovery approach. Then you go for a preclinical development and identify 250 compounds. And next it will take around 3 to 5 years. The next phase is going to be the clinical development phase, where various stages. Zero stage will be the effect, studying the effect of the compound in the body. Then you study the safety in the humans. And finally, the effectiveness and the effectiveness at treating the disease will be studied. So this is the stage 2. So for all this process, it takes 5 to 7 years. So for clinical development, it is around 5 to 7 years. Then after clinical development itself, you have other phase 3 and phase 4, where you have a large-scale safety and effectiveness process and long-term safety. This also should be passed out by this compounds, and finally the one compound will be taken for the regulatory approval, and it will be approved by the FDA, and finally it will come into the market. So totally, if you see that it takes about 15 years for a drug from a basic research to a development of a drug. So totally 15 years of timeline is required for particular drug. But this process, if you see, we all researchers stop at this level where we do our PhD and we identify a suitable compound, and before taking into this, we stop it here. So most of the time it is stopping here. So getting into this, it totally takes 15 years of timeline. So next application is polypharmacology based studies. In polypharmacology based studies, one drug, previously it was like one drug and one protein concept or one particular target molecule to be focused. But now it is like polypharmacology based studies, where you can take 2 or 3 drugs and focus on a multiple target. So this type of studies, you call it as polypharmacological studies, where you have you take the drugs and you study with multiple proteins. So one drug with multiple proteins, and you study there where they are changing the metabolic pathways are being affected, their biological interactions are being affected, and then finally you screen the drugs for their responsiveness with the phenotypes. So how and where the essential pathways can be inhibited by this particular drug for treatment during the disease process can be identified, and also the safety of this multi-target drugs for further combinations can also be revealed using polypharmacology studies. So this pharmacological interactions, then you study about the biological interactions, then drug combination screens will be taken. So when nowadays we see that when we are going for a particular treatment, they give us 2 or 3 combinations of drugs, right? So such kind of polypharmacological studies are helping to screen such type of combinatorial studies. Next one is polypharmacological studies. This is one example. You are consuming one drug, the metabolites gets activated, and similarly the compound, drug interacts with the compound interacts with the target molecule. So when it is interacting with the target molecule, all those 2 networks are combined, and you get a metabolite target network. So this type of studies, you call it as polypharmacological studies. This is the overall view of how the polypharmacological network is being created. Next one is the enzyme substrate interaction. So enzyme substrate interaction, it also comes here. where you use molecular docking approach, where you investigate their binding modes of the substrates to the enzymes, and you can understand the catalytic mechanisms, and you can predict the mutations. Say, for example, DNA gyrase is having an QRDR region. This QRDR region, you call it as quinone resistance determining region. So in this particular region, your fluoroquinone drug goes and binds, and this particular region is specific for targeting a particular type of an organism which is causing infection in humans. Say, for example, it is a bacterial organism. So this QRDR, when this organism is very intelligent, it keeps on creating mutations at this, because when this drug is going keep on binding to it, it creates a mutation in that. So this type of mutations can also be studied using molecular docking studies, where you can identify that particular target sequence, then you go for a modeling, then you can find out the mutations present in it. Next, you can study about the protein-protein interaction studies. In protein-protein interaction, as I mentioned previously, either you can study the interaction, you can see one drug is regulating other proteins, or what are all the proteins which are being regulated, and you can understand the whole cellular signaling as well as the regulatory networks. Or you can create a mutation in a particular site, or you can target a particular protein which is involved in the network, so that the whole network will be collapsed. So like that, you can design your work. Next one is biochemical mechanisms. So you're understanding how the ligand modulates the protein function, and that could also be in a design for targeted therapeutic applications. So next one is structural biology insight, where this is like you can design the experiments and interpret the experiments based on the experimental data which is already being present. So most of the structural data can be validated. Next one is predicting binding affinities. So here, if you are here it is going to be the binding affinities, where your lead molecules are showing specific binding interactions. So how far they are being having binding affinities with a particular protein can be identified, and that protein can be prioritized or taken to the preclinical research. Next one is in silico toxicology. In silico toxicology based studies, you can study the safer drug candidates based how the pharmacokinetic properties of the particular lead molecules or there can be investigated, and that can be further taken for molecular docking studies, and it can be taken for further clinical diagnostic studies. Next one is optimizing protein engineering, where the novel proteins or the enzymes with the desired function can be identified, and their mutations created at that assigned mutations at that particular site of protein ligand interactions can also be studied. Thereby the optimization can be done, so that next level of enzyme screening can be done, so the high level of enzyme production can be enhanced. So all these types of ideologies can be induced when you are doing such kind of molecular docking based studies. So it is also used in personalized medicine in order to study how the specific genetic variations may influence a drug's response. So next important topic is the drug repurposing or approaches. You can see that it starts with molecular docking, and there is a series of steps in discovery process. That is, it starts with molecular docking, then transcriptional signatures, similarity analysis, network analysis, machine learning, and data mining. Whereas in validation step, it starts with knockdown signature, meta-analysis, cell culture studies, protein signatures, xenografts, and ends up with molecular docking. However, in drug repurposing, you use molecular docking even at the starting stage or at the ending stage. Either or the other way, molecular docking is an important procedure. It is being applied in drug repurposing approaches. So the general chemicals, if you see that the drug that is fluoroquinone and hydroxyquinone, it was used in anti-malarial medications, but it was also used for autoimmune diseases. It means that one particular therapeutic application already it has been approved, but still it is being used for when the situation is where there is an outbreak like COVID, it was being given for studying whether there is any reduction in that particular diseases or it's having any therapeutic applications. Similarly, FDA approved drugs can be used One drug is the psychotropic drug, that is the chlorochromomazine, which is evidenced to have some anti-proliferative activity against colon and brain tumors. So this drug is a psychopathic drug, but it was also given for colon and brain tumors. So this is the methodology or the steps which is followed in drug repurposing, where you have a computational screening data. This drug, either it will have a new, it can be used for a new target, old indications, or new target, new indication, or old target, new indication. So this drug can be taken for different aspects. Then in silico screening can be done. After doing the in silico screening with molecular docking or virtual screening, then it can be taken for in vitro screening. The topmost compounds can be taken for in vitro screening, then in vivo screening, where preclinical screenings can also be carried out. Then after in vivo screening, it is taken for clinical trials, and finally it is given for FDA approval. And this FDA approval, if you see, it has already been drug is being approved, and this successful drug can be repurposed for other therapeutic approaches. This is the general procedure, but the same thing can be used for the FDA approved drug. So the drug, the drug target interaction, changes in the metabolic pathway or the gene expression, and finally changes in the phenotype. Anyways, it has been approved by the FDA for safety indication. So already it is safe. It has been given for safety indication, right? So it can be used for successfully for repurposing of the drug for various therapeutic other indications. So what are all the receptors or the proteins that is being involved in molecular docking? So you have a protein, it can interact with a ligand molecule. You can see that molecular docking can occur between a protein, this is a protein, and this is your ligand. So it can occur between protein and a nucleic acid. It could be a DNA or the RNA. So it can occur between a protein and a protein. So you can see a network of proteins showing interaction. So molecular docking can occur between these types of moieties. So next we are moving on to steps in molecular docking. So we start with the major basic steps, that ligand and receptor preparation. So this is the first and the foremost step which we need to focus. So when you download a very good protein structure, then it is going to be well and good. Otherwise, you have to keep on validating your results. Sometimes it may. So preparation is the first part where you have to focus with, and similarly for the ligands. So for if the 3D structures for the ligands and the proteins are available, then it is fine. If it is not available, what we are supposed to do? So ligand preparation is the first step, and the protein receptor preparation is also added to it. Next one is the grid generation. So grid generation is nothing but a grid which is kept on the receptor. So we can visualize these see tomorrow in the demonstration session. But we'll see the steps and we'll see the elaboration or what we are going to do one by one. So next one is grid generation. It is like a 3D grid which is kept on the receptor to the specifically to the binding site or the search space where your ligand will go and bind to the particular site. Next one is the scoring function, search and docking calculations, scoring and ranking. So all these things will be given in your docking software. But you have to understand the terminologies. I'll explain one by one in the next slides what are all these terminologies and what you have to focus. Because only if you know these functions, it will be easy for you to write the research article or your manuscript. If you're preparing for a dissertation, it will be very useful for you. So next one is scoring and ranking. Next one is analysis, visualization, validation. Then you are supposed to optimize or refine your results. And finally, you go for reporting. So these are the basic 10 steps which you are supposed to follow in molecular docking. So one by one, we will see what are all the steps. So first one is ligand and the receptor preparation. First step is the ligand preparation. So when you have a ligand, say for example, you are doing a wet lab experiment where you have identified a particular compound is showing interaction with a particular, you are doing a qPCR, using qPCR, you identify the gene is being overexpressed or it is being suppressed when you given particular compound. So this compound you have either isolated or you have retrieved from some source. Okay, so this source you have the structure, whether you have the structure of the particular ligand. If you don't have the structure, you can draw the structure if you know the structure, or if the name of the structure ligand is known, you can download it from some common databases. Those databases such as example PubChem databases or ChemEMBL, you can download the structure of the database and you can save it in the system. Next one is, if you are not able to prepare, you have to prepare the ligand in such a way that they are able to be in a, with the correct moieties or charges being applied, so that they will have an interaction. So for the molecular modeling environment, you have to prepare that particular ligand. So we use OpenBabel or Pybel in order to prepare this molecular modeling. It is also a molecular modeling software where you use for preparing your ligand structures. And also this JMOL, PyMol, it is a visualization tool where you can open and visualize your prepared ligands. Then it is the then you can use quantum chemistry softwares in order to add some charges, or if you want to do some optimization or refinement, you can go for such softwares. So next one is receptor preparation. If you have a protein structure, that is your receptor, it is well and good. If you don't have the receptor, so if you have the receptor, you know the protein name. Say for example, you have a DNA guidance, you download from with the protein number, you will just download from the PDB data bank, that is a protein data bank. So from PDB, download and store it in your system. If you don't have the model, you can go for homology modeling. That is, you can either use web servers or standalone. That is, modeler software is available. You can download, install, do the modeling. Then you can go for molecular docking. So then you do the online modeling using Swiss model, or you can already homology modeling database is there. You can directly download the models from the Modbase. Then you download it and you keep it in your system. Then you can either go for refinement of your or optimization of your protein or the receptor using some molecular dynamic software, that is GROMACS, AMBER or CHARM. Then after optimization, you can prepare the structures which is going to be suitable for this molecular docking using this type of tools. Or you in molecular modeling tool itself, you have this molecular preparation opportunity where Autodoc, Vienna and Glide, they prepare the molecular docking of your environment for the particular receptor protein. Then you have some visualization tools. You can visualize using PyMol, Chimera. Chimera also does the receptor preparation. That is, you have the option as doc prep, where you can prepare your ligand and the receptor for suitable for molecular docking. Once this is done properly, then you have grid generation. So grid is it is a 3D three-dimensional setup where you exactly keep it on the binding site of the receptor. So why we are doing this grid generation is in order to explore the possible binding orientation of that particular ligand which you are going to study further. So you are providing a space or the environment for grid generation. So the steps involved here is, first you have to define the binding site, then you have to set the grid dimensions, then grid spacing has to be given, then you have a grid center, then generate a grid, and you can visualize the grid. For all these things, if you want to adjust the parameters, you can adjust. But if you say, for example, you are working on a human serum albumin. For human serum albumin, if somebody has done the research work and it is published in some paper with that particular grid box, grid spacing is already available in that research article, and only you are going to change the compound or the ligand, then what you can do is, you want that particular site to be focused. Then you can use the same dimensions, and you can use it in the molecular docking. Generate a grid for that binding site, and then you can allow your ligands to bind to it. Or if you are going to study on a different site based on your experimental results, then you can use a different parameter for that, and or else you can adjust your parameters based on your requirement. So these are the basic steps you have to understand, and just the terminologies you have to go through. So this binding site, dimension, spacing, center point. So all these things you have to include in your manuscript or your report, whatever you're going to prepare for them. So next one is scoring function. Scoring function it is used to quantify the binding affinity. So we have already seen that binding affinity is a measure of measure of particular intensity between the ligand and the receptor when it is showing interaction. Likewise, they give a quantity or a quantify that binding affinity. So that function you call it a scoring function. So what are all the key aspects is energy calculation. So how much energy it is being used for a particular ligand force. That function you will be collecting. Next one is the force field. So force field here it will be a set of physical forces acting between the ligand and the receptor. So you have a set of parameters defined here. So you have to remember this when you are doing your dissertation or your manuscript. You have to mention this type of bond length, bond angle, dihedral angles, non-bonded interactions. All these force fields and their output values can be provided. So it will be very effective to understand the bond length, how far the binding affinity of each and every ligand to the receptor it is. So you have to remember these terminologies when you are doing your manuscript. So these terminologies you have, that's why I'm telling you all these things. Next one is your empirical and the knowledge-based scoring method, where you rely mainly on the mathematical expressions, where your experimental data or the knowledge-based information will be used, or the statistical information regarding the ligand and the receptor complex will be focused. So these are the common terminologies in scoring function. This is van der Waals interactions. So it means that there is a repulsive force or the attractive force between the atoms. So this is denoted as van der Waals interaction. Next one is the electrostatic interaction, and it mainly accounts for the interaction between the charged atoms. Next one is hydrogen bonding. So it evaluates the bond formation between specific atom pairs. Then desolvation energy, it considers the cost of displacing the water molecules from the binding site. So these terminologies, the scoring is also being calculated for all these forces, both you have seen here as non-bonded interactions, and these are the bonded interactions. So for all these things, the scoring will be applied, and for all these things, you have to include the terminologies in your manuscript or the report which you are going to prepare. So it will be very effective when you know these functions. So when you are preparing your validation report, it will be easy for you. So next one is your scoring function. So in scoring function, one particular terminology is weighting. So weighting means it assigns a weight to the different terms based on the ligand receptor interaction, and it when you adjust the weight, it can have a heavy impact on your scoring as well as ranking of different ligand forces. So range here, it is going to be the range between the specific scale, and you can understand the scoring range. It can correlate with your binding affinity. So finally, you will be doing the validation and optimization, where machine learning approaches are also being used. When you have when you are doing this scoring function, when you are doing for only one or two ligands, it is very easy to do. But when you have a large data set of compounds, then you need a scoring function. So always when you are having a large data set, this scoring function is going to play a very vast role. So it will help us. It is a machine learning techniques or approaches are also being used to tell us how the complex pattern from a large data set of the ligand or the receptor is having an interaction. So for one single protein, you take a database of having 1,000 molecules or showing interaction. Then you cannot, you manually cannot sit and do, right? So you need a machine learning technique where a scoring function is going to play a major role. So similarly, search algorithms. So what are all the search algorithms it is being used? One is random search, grid-based search, genetic algorithms, Monte Carlo methods, and Lamarckian genetic algorithms. So random searches to keep a ligand binding to all the sites anywhere randomly to the protein. In grid-based search, where you specify a grid to the particular protein, then you allow the ligand to interact with that particular inside the grid. Next, you use genetic algorithms where you use crossing over or mutations that to evolve around the ligand conformations. Then you have Monte Carlo methods where it will be using energy criteria for random sampling of the ligands. Lamarckian genetic algorithms where you use a combination of both the genetic algorithms with the local optimization in order to refine your ligand pools. These are the general search algorithms which has been applied. So you can just have a look on it. Then finally, you come to the docking calculations, but all these things are being calculated in your software. You need not worry about it, but still you have to know about these terminologies, because when you are preparing your manuscript or your paper or your whatever thesis or what, then you are going to read some articles, right? Then you have to know the terminologies. That's why I'm telling you all the terminologies which I find to be essential for you. So how the docking calculations is being calculated is, you first make a grid-based placement, then conformational sampling will be done, then energy evaluation will be done, then scoring functions will be applied, then ranking of the poses will be carried out, then output generation and visualization will be done, and finally the analysis and validation. You are not going to do any of these things. It is just a terminology you are going to understand, because your software is going to do all these things. So, but when you're going to do an in-depth study, then you are supposed to know about all these things. Next one is scoring and ranking. So, scoring, as we know that for each orientation and conformation, your ligand showing interaction with the receptor based on the affinity, it is going to get a score. And in ranking, how the rank is going to be given is based on the score. The ranking is going to be allotted. For some tools, you give an option that I need only 10 ligand poses. So based on that, ranking will be given. The topmost ligand, which is showing the lowest least score, will be considered to be most energetically favorable. So such kind of ligands will be given the top score. So that is the rank one of the ligand. And then if you are seeing that there are several types of forces, such as van der Waals interaction, electrostatic interactions, hydrogen bonding, and solvation energy. All these scoring individual concepts will be generated, and that also will be coming out in the software, along with the software, output in the software. Next, you can visualize and analyze the hydrogen bonds present, number of hydrogen bonds present, what are all the key interactions at the atomic level, hydrophobic contacts. All these contents can be understood. Next one is the post-docking analysis. In post-docking analysis, you will be studying and predicting the binding modes with the experimental data or the crystallographic data, which is whatever has been analyzed. You just compare your post data, validate your data with the experimental data. Then you can do some iterative refinement. Once you understand your docking results, then if you want to adjust the parameters, you can go adjust and refine the setup. Again, you can do the docking procedure for improving the accuracy of your predictions. And finally, you select the top rank the binding pose, and finally you take it for biologically relevant, and you find it to be biologically relevant, you take it for further preclinical or clinical studies. Finally, you use several web servers for analysis and visualization. These are the web servers which is widely being used. One is the ProDigi server. Using this ProDigi server, you can calculate the binding affinity, and you can also assess the protein-protein interaction. And this using PDB sum, their molecular structures can be studied, and you can also visualize your docking result. So from LigPlot, you can generate the protein ligand interactions, and you can also visualize the hydrogen bonds and hydrophobic contacts. Then you have a BioBR Discovery Studio Visualizer. If this tool is a standalone tool available, and you can download this and you can install in your system also, and you can use for varying analysis of your docking results. Next one is PostView. It is also used for studying the binding poses and interactions. And this PDB, the protein interface, surface, and assemblies, all these concepts are being used for studying in the protein ligand interaction output result. This Caver analysis is very, very useful, where you can see the tunnels and channels in the protein. So ion gated channels, voltage gated channels. So for all these things, you can use this Caver analysis. And protein ligand interaction profiler, it is also doing the same function. So these are the standalone tools for visualization: PyMol, DMD, UCSF Chimera, Visualizer, Autodoc tools, Chimera X, Mastro, that is from Schrodinger, and OpenBabel. So these are the standalone tools. Next, for validation, we have some web servers and standalone. So most of them do not know how to validate our results. So you can either download the standalone tools and you can go for validation, or you can directly submit your protein ligand complex to the web servers, and your it will compare your data with your predicted data with your experimental data, and the binding energies will be calculated, and your data will be your results will be obtained in your email ID. This HADOF also does the same thing, where it is mainly it is used for improving the accuracy of your predictions. Then Cluster Pro, it is also enabling us to validate and improve the docking results. And in standalone, Ligand Scout is it is also useful for predicting binding forces and experimental data. In Vienna software already we know that it can directly give us the show us the experimental results of docking procedures. So finally, if you want to do some optimization and refinement of your molecular docking studies, you have to go for molecular dynamic studies. Molecular dynamic simulation, it is one of the refinement of in your docked complexes, where in our human system, you have a protein which is at 37 degrees centigrade. The same system, same um, environment will be provided in molecular dynamics simulation to in order to study how far your protein is having and structure when it is interacting with the ligand molecule. Such type of environment is provided and validated. So this it is a mimic of your human system in an computer. So it will help us to refine our molecular docking results. So molecular dynamics is one of the important refinement programs. It is using various tools such as GROMACS, AMBER, NAMD. So these are the dynamics simulation softwares. And then you have for energy minimization, you can use this optimization energy system tools such as CHARM, GROMACS, and AMBER. It will help us to study the atomic coordinates where it is interacting, how iteratively it is adjusting with the ligand moieties. So all these things can be studied using energy minimization. Monte Carlo simulations can be done. It is a paid version of Gold and Autodoc. They use this popular tool for doing Monte Carlo simulations. And similarly, Schrodinger soup is also Glide is also a hybrid method where it is using molecular docking as well as dynamic molecular dynamic simulation, where it uses quantum mechanic approaches. So likewise, we have several optimization and refinement tools for your understanding. And next, final part is going to be your report generation. So in report generation, what are all the steps? So you have to document your final ligand process. So how the documentation is done and what basis it is being done. First, you have to include your receptor and the ligand interaction. So how they are being included. The first step is going to be your scores and ranking. So what are those scores assigned for this particular docking pose, and the rank for the pose based on their scoring function.
அட்டன் பண்ணி
Please wait for a second, sir.
So what happened, sir? Madam is left.
I think so. Some technical issues, sir. Please wait for a second, sir.
Sorry, my. So where are we actually? Where my screen got cut actually? Did you see this steps in report generation,
Yes,
right?
ma'am. Yes, ma'am. Yes, ma'am.
So the next one was drug identified through molecular docking. So these are the drugs which are being identified using molecular docking. So the virtual environment came into two where the drugs are being screened against various types of molecules. That is, these are the enzymes. Neuraminidase is an enzyme which is present in influenza virus, where you can treat and prevent influenza using this particular drug. Then you have imatinib. It is also targeting tyrosine kinase, and where it is chronic myeloid leukemia is being prevented. So Ibestar, this is the brand name for estrogen receptor for preventing osteoporosis and breast cancer in females. Next one is Retias and Carleta, where these two are protease, HIV protease inhibitors, where HIV can be prevented. So this is how molecular docking has played a major role in identifying suitable drugs against a particular candidate, and it is used for prevention and treatment. So the take-home message for today's study is you have a database. When you identify a receptor molecule here, so receptor, if you know it is okay, you can directly download it from the database, that is the PDB database. Then if you don't have, you can go for homology modeling. Then or else you can download it from the modbase. Then you can keep it or save it in your system. Then you have your ligand. So ligand, similarly, if you know the structure, you can draw the structure using ChemDraw or any drawing software. You can draw and you keep it with you. Then you can go for refinement and then you can store it in your computer. Next, you can use this molecular docking tools and do the molecular docking between the ligand and the receptor. The output will be your results, and these results can be validated, and the based on their binding mode, scoring function, and their interactions. All these reports results can be represented in your final report generation, and it can go for experimental validation. And the same vice versa can be followed. That is, when you have a experimental data, you can take the experimental data where you can identify the particular ligand or the protein, and then you can proceed with and you can validate your results. So this is your take-home message. And tomorrow we will see how to do the demonstration using an tool and how to visualize your results. So thank you for today. And any questions you can ask me now.
Ma'am, I wanna ask one question. Am I audible?
Yeah, tell me.
Actually, I want to ask on the drug designing background that if you want to search any tool regarding the perfect concentration which is required for that compound in the body, then how can we search that tool for that? Which concentration of a compound is required in our body or not?
In
Otherwise,
pharma,
yeah.
there is a way, but that is also a prediction. We cannot directly, or we cannot say that this is going to be the exact rule. But the IC50 value for a particular concentration of a particular drug can be revealed. So the IC50 can be revealed using the online servers. There are so many online servers for predicting this IC50 value of a particular drug molecule. So you can give the chemical structure and it will give the concentration of a particular drug to be used. And there are also some paid version softwares which are available with some companies where you can approach and you can ask them for help for doing this type of research work.
Ma'am, can you name some free software? Then it would be helpful.
Free software, you can see web servers. You can just paste as IC50 values freeware. You will get a list of IC50 calc, I think. That is also available.
Okay, ma'am.
Hello,
Thank
am I
you.
audible, madam?
Yes, sir.
Madam, can you take it hands-on in tomorrow?
Yes, sir. Tomorrow I will be showing the molecular docking, how to do a molecular docking, how to download the proteins, ligands, store it in your computer and do a molecular docking. And then we can validate the results, take the output, and you can understand the how the representation has to be made. So using a visualizer. So likewise, we'll have a demo session, sir, tomorrow.
Okay. Thank you, madam.
Hello, ma'am.
Thank you. Yes.
Hello, ma'am. You told in one of your slide that we have web servers like deep site for finding our active site. Okay.
Yes.
So, are there any server or software for finding the allosteric site, ma'am?
Allosteric sites.
Yes, ma'am.
Exactly, I am not aware of this, but allosteric sites, we can just read the research article and we can check. They will not directly give you the name as allosteric, but there could be some hidden names. For that, what we can do is we can just read the articles and find out what would be the term, and then we can go for such types of tools. So,
And
sir, yeah, right.
ma'am, can we just predict allosteric site by like doing molecular dynamics because it will show the dynamic nature of the protein? Can we predict like?
No, I am not clear with your question. Can you repeat it again?
So my question is, ma'am, can we predict allosteric sites by molecular dynamics?
Molecular dynamics, allosteric sites. I don't think so. We can do that.
Okay, ma'am. Thank you, ma'am.
Thank you. Yes, tomorrow will be a molecular docking visualization tool. We'll be having a demonstration with that.
Ma'am, you told me there's so many softwares now, ma'am. Each
Yes,
and every step. Is it all are freeware or it's a paid version?
Yes,
ma'am?
most all of them or whatever I have shown is like it will be useful for everyone. Like it is all are freewares only. So there are so many web servers also to validate our results. So most of them, whatever I have shown you is an freeware.
Okay, ma'am, sure.
Except this Mastro and some other tools. In that also as a visualizer, they provide us a freeware.
Okay, ma'am.
Yeah.
Thank you.
Thank you, ma'am. Thank you so much for your informative session. And thank you, participants. Let's meet in the tomorrow's session. Thank you, one and all.
Thank you, Nandini. Thank you.
Thank you, ma'am.